So we're talking about visual pathways, and I actually looked at the syllabus and realized that we got information to the visual cortex on the schedule and then moved right into objects and scenes, perceiving objects and scenes. And we really need to put those features back together into objects again. The brain needs to do that before we can perceive anything but features. So we really need to get up into this higher level processing. So if we do this, how did we get here? Light hit the photoreceptors, went through the bipolar and retinal ganglion cells. Don't forget the horizontal and amacrine cells. That information got sent to the lateral geniculate nucleus of the thalamus and the superior colliculus. The LGN sent that information on to the striate cortex. Superior colliculus, that's going to help us with eye movements, so another subcortical structure. And the question is now, from the striate cortex, or V1, primary visual cortex, where does it go from here for higher level processing? So once information gets to the striate cortex and gets sent beyond the striate cortex, it takes two primary pathways. One dorsally. So if you think of the dorsal fin of a shark towards our back, but because we're bipedal, it's up towards the top of the brain. And this goes through the parietal lobes, which we call the where or how pathway. The other pathway goes ventrally, which is down in the brain, towards the temporal lobes, those lobes that are like above the temples, and we call this the what pathway. I'm going to present some of this research for how we discovered these pathways of vision. This comes from Ungerleider and Michigan in 1982. And this is an ablation or lesioning study. Really, it's an ablation study because they're removing this part of the animal's brain. But so the way these work is first we test the animal's capacity, train the animal, measure perception. Then we go in and ablate or remove a particular area of the brain and leave the rest intact. And then we retrain the animal to determine which of those perceptual capacities remain and which are impaired. So for the Ungerleider and Michigan work, how this worked was they presented monkeys with two tasks, an object discrimination problem and a landmark discrimination problem. So in the object discrimination problem, they are distinguishing between two different objects. There's a cylinder, let's say, and a block. And every time the monkey looks under the block, there's food. So they're getting reinforced and conditioned to look under the block, and that's what they're learning. The landmark discrimination problem is more like there are two wells, and they're both covered, and there's one with a cylinder or some kind of landmark next to it, and so that's considered a more of a where task, right? As I'm looking for the landmark to tell me where the food is, and the food is in the well next to the cylinder. So in the object discrimination task, the monkeys could learn the objects. They could choose between the two objects, where choosing the correct object means they get their food reward. Here again, I'm going to say it's that rectangular block. And if then the researchers went in and took out the temporal lobes, now the monkeys could no longer do that task. And again, temporal lobes, that's our ventral pathway. And I'm just using some word art and shapes to try and help you remember that that's going down from the occipital lobes down and forward. And I know this seems like overkill, but it feels like people really didn't remember these associations, so I tried to do it with as few words on a slide. So ventral pathway through the temporal lobes, this is our what pathway. We might call it our object recognition pathway. They train them on the landmark discrimination task. So monkeys learn to choose the food well that is closest to some landmark. Again, in this case, it's this cylinder. Choosing the correct well, again, they get their food reward, so they're learning where their food is. And now they go in and take out the monkey's parietal lobes, and now the monkey can no longer do the landmark discrimination task. Okay, and again, just to remind you, this is the dorsal pathway going up and forward from the occipital lobes into the parietal lobes. And here again, just putting it together with as few kind of words as possible to help you associate them in your mind as much as possible. The dorsal pathway goes up through the parietal lobes. We really think of this as our where pathway, or how we navigate through space. And here is the picture from your textbook that I think is still in there, that has a summary all in one picture. We've gotten it back to the striate cortex. It goes up dorsally through the parietal lobes through the where-how pathway, down through the temporal lobes, through the ventrally through the what pathway. And as we are looking at that where pathway, the question that Milner and Goodale had, starting in about 1995 is when we see their first work with their patient. Should these be called the what and where pathways, or should they be called the what and how pathways? So we're going to look at some evidence that maybe this pathway, this dorsal pathway through the parietal lobes, is not just about locating objects and where objects are, but how we navigate through space and how we interact with our environment. And their argument was that it involves taking action, okay, which requires a where component. To take action and interact with my environment, I need to be able to locate and see where things are. But it also involves physical interaction with objects. So it's a level of complexity above the kind of where pathway or where component. And one of my questions when I first started teaching this class, one of my extra credit questions was, how would my friends and I are interested in distinguishing between whether this is a where or a how pathway, and come up with a study to kind of examine that. And it's really kind of difficult because they are so intertwined with each other. They're so reliant on each other. I need to know where things are to interact with my environment. So in one study, they found evidence that they took as evidence for this being a how pathway, where the parietal cortex is responding when a monkey looks at an object, and also responding when the monkey reaches toward an object. And I asked, does this answer our question? You can think about, if you agree, you might want to pause it and think about whether or not this is suggesting a how pathway. That, again, is certainly how the researchers interpreted that outcome. But I kind of feel like I'm not sure it really does answer the question. And we're going to look at some more evidence with people who have some damage to their parietal pathway. But it's interesting. I feel like when the parietal cortex is responding when the monkey is reaching toward an object, that location, that where information is just confounded with the how interacting with the world. So we're going to look at some evidence from neuropsychology. Neuropsychology is the study of behavioral effects of brain damage in humans. And to start this discussion, I usually start with this concept of a dissociation, because this is explained in your textbook, and it's a really important concept when it comes to if you're going into any field that involves neuroscience. So a dissociation is a situation where one function is present while another function is absent. And we're going to look at single dissociations and double dissociations and what they tell us about those functions and how they're represented in the brain. So if we have a single patient, and I'm going to give these first two examples are just that, they're just examples. So we have a patient, Alice, and she has damage to her temporal lobes. She has difficulty naming and not difficulty locating objects. And so, as I said, the temporal lobes are the what pathway, and so naming objects is difficult, but her parietal lobes are intact, so she has no difficulty locating. With this, what we call a single dissociation, we can conclude that the two functions involve different mechanisms in the brain, but we don't know if those mechanisms operate independently of each other, because clearly Alice can locate without having to name. And so we know these are different mechanisms, but it could be that in order to name something, she needs to be able to locate it. So we don't know if these are absolutely independent of each other, because it could be that naming depends on locating. For us to say we have a double dissociation, we need at least 2 patients with differing damage. So I'm keeping Alice up there and sort of hopefully in memory, and she's real small up there. Temporal lobe damage, difficulty naming, but not locating. And we also have Bert, okay, who has damage to his parietal lobes. He has difficulty locating, but no difficulty naming. And now, so we can conclude that these 2 functions involve different mechanisms and that these mechanisms operate independently of each other. Not only can Alice locate without having to name an object, but Bert can name an object. He doesn't necessarily need to locate the object. So we can show this complete independence with a double dissociation. So we're going to start by looking at patient DF. They usually use their initials, these patients, to maintain their privacy, but also to distinguish them from one another. And if my memory serves, Milner and Goodale, I think they have quite a few papers where they are running DF through various tasks. She has damage to her ventral pathway. She is not able to match the orientation of a card held in her hand to different orientations on a slot. So if we're looking at A in that picture, we have a vertical slot, right? And with the control, so this is a, quote, normal person, when we're trying to match the orientation, we're pretty close to vertical, right? It seems like a pretty easy task if your brain is intact. Whereas DF, she's matching to a vertical slot, and you can see all of her different tries and trials that she is at various levels of oblique and even sometimes completely horizontal in trying to match that orientation. However, if they ask her to mail the card through the slot, she can place the card into the slot. And then so in B, we're showing the control person that we again are very close to vertical. DF, the active kind of posting, as she's putting that card into the slot, she's pretty close as well. So we have this conclusion of we have one mechanism for judging the orientation of line, another mechanism for coordinating vision and action to do something with that orientation. And again, Milner and Goodale really interpreted this as a how mechanism because we are coordinating vision and action. So with DF, we found one mechanism was for judging orientation, another mechanism for coordinating that visual information with some kind of action. And I have this question here, green question means you're supposed to think about it, right? In face-to-face classes, we'd be trying to answer this, but are these mechanisms independent of each other? Do we know this from DF? And I think you might be able to tell from my next green question that the answer is no. We don't know, right? How do we find out? And again, I'm going to suggest that you pause and think about that if it didn't just pop into your head. Okay, and what we need is what we actually end up finding, that people who have damage to their dorsal stream, some of these people, depending on the damage, can judge the visual orientation, but then when they go to mail the card through the slot to accomplish that task that combines vision and action, they're not able to do that task. So these mechanisms are independent of each other. We now have the double dissociation. And I come back to this question of does this start to sound more like a how, where the dorsal pathway is now thought of as a how or an action pathway, at least as much as a where pathway. And again, you can think about how you feel about that evidence. Other evidence about this pathway, we have evidence from people without brain damage. They've used this visual illusion, a visual illusion. And something that when we were making the judgments, we know that there's an illusion, but then they also had people do this kind of reaching and grasping task, and you can kind of see that they had these electrodes on their fingers so that they could measure exactly how much they were separating their fingers in order to grab or grasp these lines. So in A, what's happening is you're just saying which one appears longer, line 1 or line 2. And for most of us, we have this illusion that line 2 appears to be longer. It is taking up just about the same amount of retinal space, but it has this illusion of being farther away because they've given us this depth cue to show that it's farther away. And so because it's taking up as much retinal space, but it's farther away, we interpret it as being larger. If we look at the segment in B, in that image, what they're showing is the actual length of the lines. That line 1 is just slightly larger than line 2, just a little bit longer. Now, if people are making that estimation, and here they are just, they're moving their fingers, but they're not reaching towards it, they're just making an estimation. You can see the length estimations down in the figure in D. They're saying line 1 is slightly shorter than line 2. But with that same illusion, if we have them go to grasp for so to move their fingers as if they're going to grasp that line, their grasping has line 1 as being longer than line 2, which is appropriate to reality. So again, suggesting that this illusion is really affecting the what pathway, or the putting together of visual information into objects and what's out there, but not influencing so much the how pathway and interacting with the world. And I think I have a few bits of evidence here of how when we get beyond feature detectors, as I keep calling those simple cells, complex cells, and end-stop cells, they're just detecting features, but we want to get beyond that. Charles Gross and his colleagues back in 1972 were looking at the infratemporal area. So if you know, this is the inferior area, so the lower part of the temporal lobe, so the IT area. And this is where we're doing some of our object recognition. They're looking at monkeys, and it was another one of those serendipitous kind of studies or findings where they were using dots and lines and things, thinking that that's what the infratemporal area was going to respond to. And then one day, when the monkey was hooked up still, the researcher pointed at something, and his or her, I can't remember, hand went across the screen, like had the shadow on the screen, and that's when those neurons fired. And they realized that this was it's really kind of a hand neuron, right, responding to the shape of a hand. And what we see is, as we're out at circles, squares, or lines, we have no neural firing. As we move into other objects that might have some finger type shapes, kinda hinting at them, we have a little bit of neural responding. And as we get more towards hands, we have our maximum responding. And you can see really in 6, the maximum responding is to a monkey's paw, to a monkey's hand. Similarly, Tanaka et al. In 91 and 95 found cells in the temporal cortex that respond best to complex stimuli. I sometimes call this the lollipop neuron, although I'm sure that's not what they call it. But so what we're showing in A, so at that bottom part is the amount of neural firing. And so in A, that little peak that comes up, that's the neuron impulses per second, the neuron firing quickly to what looks like a lollipop. If what was presented was the bar or the stick of the lollipop, in B, we see almost no neural firing. If what we present is just a circle, or maybe the top of a lollipop, we see almost no neural firing. And if what we present is a lollipop, but a square lollipop, if you can see it, on my screen it's pretty clear, and when I show it in classes it's really not, but you might be able to tell there's neural firing, but not at the same level as for the circle lollipop. So we are starting to now respond to more complex stimuli. And cells that respond best to similar complex stimuli, or kind of complex stimuli that are in a category, tend to be grouped together in one area. And so we talk about the modularity of processing, where we have some structure that is specialized to process information about a particular type of stimulus. And this is called a module. And I'm going to say, well, there's not really modularity of processing. There is processing across the brain in a lot of different areas that are active, but this is still, we have these modules that respond best to particular types of stimuli. And probably one of the more famous modules at this point is the fusiform face area, sometimes called the fusiform face gyrus. They found this in monkeys. Rolls and Tovey found this in monkeys in the inferotemporal cortex. They found that the monkey's neurons responded to best to pictures of faces. And so if you look at the top row, and we're calling these faces, and that's what the monkeys are responding. We see the firing rate of the neurons are quite high, whereas in the bottom row, these different places and flowers and things are pictures that are not faces. We see over on the right, the blue bar, there's really no neural firing at all. And that is in the inferotemporal lobe of a monkey, but for us it's called the fusiform face area, and ours is on the right side. And this is just another study from Tsao et al. 2006, so where we see response to faces, and they just break it down into other non-face categories, and we really don't see much neural responding at all to bodies or fruits or gadgets or hands or scrambled pictures. Conwisher et al. I want to say her name is Nancy. I'm actually not sure if it's Canwisher or Conwisher. Nancy Canwisher and her colleagues used fMRI to find the fusiform face area in people. And again, so we see the inferotemporal cortex in monkeys. There's this rather large area responding to faces. For people, so we're looking at from the bottom of the brain, so that's on the right side. That little area is called the fusiform face area. And we have evidence from neuropsychology from people who have brain damage. Damage to this area causes a disorder called prosopagnosia, where they have difficulty recognizing the faces of familiar people. So this is just additional evidence that this area is specialized for the perception of faces. I've watched a couple of specials talking about prosopagnosia, or I've watched one and I read some of Dr. Sacks's work, The Man Who Mistook His Wife for a Hat, and other work where he has talked about someone with prosopagnosia. And I'm going to tell a couple of stories. One of them is kind of funny, one of them's kind of sad. So I'll start with the sad one so that we end up with the funny one. So the sad one is, so this can happen, and I guess probably often happens because of stroke. And so as we get older, and this is an older man who has damage to his fusiform face area. He's not really recognizing his wife or his adult children, but he knows them, right? He has other cues to who they are, their voices, the way they move, all kinds of things. But his adult children don't live in the same town with him, and they have young children. And he doesn't really recognize his grandchildren. From time to time, from each time they see him, they change enough that he can't put together the information, and the information from their face isn't giving him anything he can recognize. So it's kind of this sad sort of, why doesn't Grandpa recognize me? A little bit more funny, but maybe not. I don't know. But as so a man and his wife were in a restaurant, and he is looking at someone and saying, that guy's staring at me. And he keeps looking up, and he keeps telling his wife, that guy's staring at me, and every time I look at him, he's glaring right at me. And his wife says, just ignore him. I mean, just let it go. And he gets more and more irate and upset. And he says, every time I look up, he's looking at me. Okay. And then he finally gets up to confront the guy, even though his wife just keeps saying, please just let it go. Says, don't cause a scene and all of that. And as he starts to get up, the other man starts to get up too. And he does this kind of take back, he's taken aback. And his wife finally turns around to look. And she realizes that he's been looking at himself in a mirror. So this can be such a profound damage. Again, it depends on the severity of the damage, but it can be such a profound damage that we wouldn't even recognize ourselves.